Hello everybody. Hello everybody and happy Thursday. So today we're going to be talking a lot about molecular genetics and I hope you guys enjoy it. So molecular genetics is the study of DNA and its function at the molecular level. So going to the nitty-gritty of the chemicals that build up and becomes the DNA or RNA. Okay. In the recent years, especially after the 1990s and early 2000s, we had a dramatic increase of data on DNA, especially because the techniques got really good. The machines that we use now to study DNA, they got fairly cheaper than started in the 80s and 90s, and they become really fast and reliable and become very easy to operate. Nowadays we have a machine called nanodrop, nanoparsor, that's like a USB-C, USB thumb, like we use in computers. And today you can do a whole genome sequencing using that. It's got fairly small, and the data processing to study these become more available as well because of the supercomputers that we all have now, and universities, they really invested in that. So the molecular genetic technology is deeply important to understand the biochemical parts of our cells and how they operate and how we can copy a DNA, how we can modify a DNA, how we can insert a gene or delete a gene. And this can translate to understanding how a cell works and their cell biology. And in a more broader sense, we can even understand how microorganisms work. We have a lot of viruses, bacteria, unicellular parasites, and it's very important to understand how they work, how they replicate if you want to combat and try to minimize the impact of these pathogens to us, understand how they can be transmitted. And everything comes from the knowledge of the molecular structure of DNA, or the deoxyribonucleic acid, or RNA, that's ribonucleic acid. And to fulfill this role, its role of the genetic material, every genetic material have to have these 4 points that we call the 4 criteria. So first of all, it has to have an information. Usually the information is built into 4 bits of information that we call nucleotides. So we have the ACTG that we're going to be talking more further in the DNA. And these have to be reliable sequence, so they had to have a sequence that is stable, they don't modify that much, they are not going to be having insertions or deletions at very regularly. So this information have to be safe and reliable. This information has to have a way to be replicated. It means that this information has to be copied over into a new cell, and also have to be copied into cells that their function is to for reproduction, that we call this process transmission. So we can pass from one multicellular organism like ourselves to our offspring. And lastly, when we pass from parents to offspring, this genetic information has to have enough variation that this new organism is different enough in a way that might have some traits that might come in handy in certain variations of its habitat, and might have some traits that make this organism less viable if it has any change of the habitat, or might even be less reliable in that habitat. But this variation is important, okay? That's the core, and otherwise we all be clones of ourselves, and one single disease would kill everybody. One single catastrophe like that would be enough to extinguish one species. So higher variation means that that species is going to have more plasticity to survive any environmental or change of their habitat, and that's enough for them to thrive in the world. If you guys ever heard about genetic bottlenecks, where you lower so much the variation that everybody is basically the same genome. So when we have anything that changes their habitat, these animals no longer, or these organisms, this could be also plants, these organisms no longer are viable, and they're going to go extinct. And that's very important. So by the 1880s, two scientists, Dr. Weizmann and Nageli, they proposed that there was a chemical substance inside the cells that was responsible for transmitting traits from parents to offspring. But at that time, no one knew what it was. It was a chemical. But that was good enough at that time. It was so good that Mendel in the early 1990s were already working with their hypothesis of their genetic of P, where color might be related to that, and so it was good enough to help us understand and help us start our first breeding programs. So help understand how we are domesticating cattle, pigs, chicken, and plants like cauliflower or lettuce, carrots. So we already knew that was something that was passing from parents to offspring, and we start select some traits for that. So for example, United States, it's very there's a breed called Longhorn, cattle breed, and started by literally selecting the male with the longest horn and the female with the longest horn, and they did this over time, and now become that animal that have that really long horn, and that's how the breed started. Okay. So we're going to be dive deep into three different experiments that were very important for us to understand how it works. So we're going to be talking about the Griffith, the Avery, and the MacLeod and McCarthy experiment, and the Hershey and Chase experiment. So the first one is a Griffith experiment. So at the time, he was working with two strings of, at the time it was called pneumococcus. Now we call Streptococcus pneumoniae or pneumonia. And they already knew that there was two strings. One that has a smooth, produce smooth colony on a solid media. Solid media is like an agar media. You guys probably studied this in microbiology. It's like a jello. And they would be smooth because they would produce a layer of sugar around the cells, around bacteria, that was made of polysaccharide, and that was called a polysaccharide capsule. And when they inject these bacteria into mice, they all would die. So they knew that they would have a high virulence. That means that they would all show disease, they would get sick, and they would also have a high mortality rate. Means most of the animals that got injected, they would die. Okay, and he was also working, and that's that strain over there. Okay, and this strain here was called rough strain, and in this strain they didn't have any polysaccharide capsule, and the colonies in the agar plate would have like a rough shape. Okay, and after injecting mice, they would not die at all. So what he did was that, so he did something called heat killing. So heat killing is one way of producing vaccine. It was well known for more than 100 years at the time, 1928, and so they kill that and use this dead bacteria to produce a vaccine. Okay, so he thought, well, what happened if I kill this bacteria using these methods of heating low and slow, and re-inject this in mice? Well, if he killed like that, probably nothing will happen. And that's so when he heat killed smooth pneumococcus, nothing happened. No mice died. All survived. Okay. But then he thought, well, if we mix a dead smooth pneumococcus with a live rough pneumococcus, what would happen? Well, in theory, nothing would happen, because a dead rough pneumococcus doesn't cause disease. A live smooth pneumococcus doesn't. A live rough pneumococcus doesn't kill. A dead smooth pneumococcus doesn't kill. So if you mix them together, in theory, no mice would die. But what happens? For some reason, the live rough pneumococcus, they start developing this capsule around them, and they start killing mice as well. So for some reason, there was something inside the dead bacteria, the dead smooth pneumococcus, that was transferring to the live rough pneumococcus, and was transforming this live rough pneumococcus into smooth pneumococcus. This process was called transformation, and until today we still call this process transformation. I did plenty of gene expressions in E. coli, Escherichia coli, where I inserted genes in E. coli, and the part where you start inserting the gene on it is still called transformation. But at that time, they didn't know anything about what was this chemical that was being transferred. They just know that was some chemical inside the cells that was transferring this information and transforming this strain of very attenuated bacteria into a very aggressive bacteria. Okay? So, in other words, I had the dead smooth bacteria that was transforming a live rough bacteria into a new live rough bacteria, because right now they would have the polysaccharide capsule around. At that time, as I said, Griffith did not know what was transforming this bacteria, but he knew that it was transforming the bacteria. E aqui just a conclusion of the experiment. So, smooth kill, rough doesn't kill. That smooth doesn't kill. When you mix that smooth with live rough, the live rough started becoming like this the smooth bacteria, and then it kills. And this process of transferring this chemical, this magical chemical, from a dead bacteria to a live bacteria, it was called transformation. Okay, and this transformed rough bacteria now have the information to make a capsule, and it was the capsule, the polysaccharide capsule, that was responsible for killing the mice. So we can see that we had two different strains of bacteria that had enough variation to make one lethal and another silly, non-lethal. One that have a polysaccharide capsule and another that doesn't have any polysaccharide capsule. But when you put that chemical that until that time they did not know, now we know that it's the DNA. This DNA was enough to transmit, okay, from a dead cell to a live cell, and becomes a new have that same trait from the dead cell. Okay, so this genetic material from the smooth pneumococcus, it was inside the cell, it was replicated within that strain, and it was being transmissible from the parents to the offspring, and from a dead parent to a live similar species. Many years passed, and now there were the group of Avery, McLeod, and McCarthy. They knew at that time, in the 1940s, what a DNA and RNA was. Still at that time, they did not know what was their function of DNA or RNA, but they at that time they were able to biochemically isolate DNA and RNA from cells. So they tried to replicate this the experiment from pneumococcus, but instead of mixing the dead smooth pneumococcus with the live rough pneumococcus, they thought, well, if now we can isolate, why not isolate the macromolecules of this bacteria and see what is transmitting from the dead to the live bacteria. So they put together in a tube isolated DNA from smooth pneumococcus, isolated RNA, and isolated proteins. Okay, and then they put in each of these tubes, they put the rough pneumococcus. They waited overnight, and the next day they look under the microscope to see if any of these rough pneumococcus have started developing the polysaccharide capsule. Okay, this time they didn't want to kill any mice. They didn't need to as well. What they observed is that only with the DNA, the rough pneumococcus would become a smooth pneumococcus. So they repeat now with just DNA extract, and some other scientists, they start criticizing how they are doing, saying because at the time the isolation of these molecules was not 100%. So they said, well, maybe there's a little bit of RNA in your DNA, or there's a little bit of protein in your DNA, and this is actually what's causing the thing that's been transmitted to the live rough pneumococcus. So they were like, okay, so this time I'm gonna be adding DNAs into the DNA extract and if there's something besides DNA that's making these rough cells into smooth cells, it's going to show up in the culture. And when they mix, nothing happens. It's still all rough pneumococcus. When they mix the DNA extract with RNAs, so they destroy all RNA from this, they have a transformation from rough cells to smooth cells. When they mix DNA extract with protease, that means enzyme that's going to break all proteins, they still have the transformation from rough cells to smooth cells. So at the end, they proved that DNA was that chemical that McHugh was studying, and that chemical is very important because that chemical have information, a genetic information, to transform a rough cell, a rough pneumococcus into a smooth pneumococcus. There is information in there to make that happen. Then in 1950s, something like 10 years later, Hershey and Chase, they also provide more information that DNA is the genetic material responsible for transmitting information to make a organism be itself. So they're again working with bacteria. This time they were working with E. coli, Escherichia coli. And there is a virus that parasitizes E. coli. There's a group of viruses that parasitize bacteria that's called bacteriophages or just phages. And these are very, very basic organisms. And by the way, viruses are now considered organisms in the last 10, 15 years. Okay. And as a very basic organism, it only has two macromolecules. It has DNA and has protein. So as you can see here in the picture, we have the capsid here that's all made of protein. And inside the capsule, in this region here, has their genetic material. In this case, bacteriophage, it is DNA. Okay? And they thought, well, we can radio label the protein and the DNA of bacteriophages and see if these phages can transmit a DNA from one strain of bacteria to another strain of bacteria. Okay? So how did they accomplish that? Well, they use a technique that we no longer use because it's super dangerous. So they inserted radioactive isotopes into the structure, the molecular structure of these bacteriophages. So for the protein, they use radioactive label sulfur, that's the 35S. So all proteins of this phage was with this radioactive isotope. And they did the same for the DNA. At the time, they already knew what DNA was made of. So they inserted a radioactive phosphorus in the mix. So all the DNA over there was radioactive as well with the phosphorus. So they just have to put in a machine to measure the radioactivity of sulfur and phosphorus, and that way they would know what was contributing to this change of transmission of information. So they have two different E. coli's. They infect those E. coli with these phages that were radio label, and at the end of it they would take all the phages from the bacteria culture, very easy to do by centrifugation, and they would see in the bacteria if they had any radioactivity from sulfur or any radioactivity from phosphorus. Okay, and what they saw was that after the centrifugation, the bacteria, they did have radioactivity from phosphorus detected in cells, while they would have no radioactivity from the proteins from the phages, meaning that just the DNA was being transmitted from the phage to the bacteria. Okay, and if they need to build more phages, they would build from the DNA that was inserted in the E. coli. So that way, proving once more that it was no protein, it was DNA that was responsible for transmission of information. So the results of Griffiths, Avery, McLeod, and McCarthy, and Hirsch and Chase were very important to conclude that DNA is the blueprint of the cell. And here we have some epithelial cell here under cell culture, and you can see that the DNA is diffusely distributed in the nucleus of the cell, and when the cell is under mitosis, it's going to be condensated into chromosomes, and these chromosomes are going to separate into two different cells. That's what this picture is showing. So let's talk about what's the molecular structure of DNA and RNA. So DNA was first discovered in 1869. It was isolated from white blood cells, nuclei, so that's why they called nucleic acids, since they do produce an acid environment by the release of ions H+, hydrogen+. It took, as you can see, almost 100 years from the discovery of the DNA as a molecule to what's the function of DNA and RNA. DNA and RNA, they are very similar in their structure and have very different functions, okay? In eukaryotes. In viruses, they can be very similar over there. So both DNA and RNA, they do have nucleotides that carry these 4 bits of information, and these nucleotides, they are linked together in a strand of nucleotides. So that's the two things that they have in common. DNA and RNA, they have a similar structure, which have nucleotides linked in a strand. Okay? Other things that are very similar, DNA and RNA, they both have 3 components. They have a phosphate group, a sugar, and a base. The grouping of these 3 becomes a nucleotide, and the grouping of sugar with a base is a nucleoside. Okay? And as we can see here in the arrows, we already have our first big difference between a DNA and an RNA. So the DNA is missing an oxygen here in the second bond here. So it gets its name, deoxyriboses, means deoxy means without an oxygen. Ribosis means this formation, this is a ribosis. While an RNA does contain this set of oxygen, and here it's just showing again, and this is going to become a big difference. So just by looking here, which one is a nucleotide, which nucleotide here is a DNA, which one is an RNA? So it's missing oxygen, so A is a DNA. Oh, it has the second oxygen, so B is an RNA, just a ribosis. Okay? Each of these bases can carry energy. If you guys remember in cell physiology, cell in your biochemistry as well, you probably heard about a molecule called ATP, adenosine triphosphate. So adenosine triphosphate is nothing more than a nucleotide, adenine, ribosis or deoxyribosis, and 3 sets of phosphate. Okay, you also have adenosine diphosphate with just 2 phosphorus, or adenosine monophosphate with just 1 phosphorus. Here we're going to talk a little bit about the structure of the chain, the DNA chain. So coming from the 5' to the 3' direction, we are going to start the 5' with the phosphorus as a starting point. Then we're going to have a ribosis, or in this case, the deoxyribosis, that is going to be connected to the next phosphorus. Okay, and in the 1' bond of the ribosis is going to be connected bases. DNA can have 4 different bases, 2 purines and 2 pyrimidines, which are the thymine, adenine, cytosine, and guanine. Okay, and these are the bits of information that's going to be useful later for producing proteins. Another feature that's very important is the structure of the DNA strand. So when we have one strand of DNA, it's going to have a complement on the other side, and this complement is very unstable and it's going to form an helix formation. And because the way that this helix formation forms becomes a double helix formation or double helix formation that was covered by Watson, Crick, and Wilkins in 1953, which they all were awarded the Nobel Prize for that. So, I like this one better because you can see the bond between the adenine with thymine and cytosine with guanine. I think that's very important, this hydrogen bond here. And in here we have the ball and stick model of DNA that's very common to see in high school, showing not only the minor growth but the major growth. And here you can see in the space filling model the same thing. And the structure is stabilized by these hydrogen bonds between the bases. And there's a rule that's the ATGC rule. Adenine always connect with thymine, guanine always connect with cytosine. G and C have a really strong bond because they have 3 hydrogen bonds occurring between them, so it's very hard to break them apart. It needs a lot of energy, a lot of heat, while AT, they have a lower energy level, it's easier to break them. And because of that, you're going to see that a lot of the starting points of polymerases It's going to be in sites where they're full of A and T. That way they require less energy to start. While the first string, we conventionally say they start from the 5' to the 3', the complementary sequence is going to be complementing this in the opposite direction. So it's going to be from the 3' to the 5'. Okay, as you can see here. So while the main one starts in the 5', the complementary one starts at the 3'. And the nucleotides are also going to be complementary. So C is going to be connected with G, A is going to be connected with T, or the other way around, and that's the law, that's the rule, and that's how it goes. DNA loves to coil, and when it's not producing any protein, it's not duplicating, they do coil around histone proteins. That's going to help it fold and compact the DNA strands into so they occupy less space, and they're not going to be producing any protein while they're in that compact form. Of course, DNA doesn't stay in that double helix form, they say in their double helix form, but the way that they stay is not stable. So the form that we found in most living cells is called B-DNA, and this B-DNA can be rearranged into a Z-DNA that's basically a mirror image of the B-DNA. And the Z-DNA is linked to transcription. So when it's forming RNA sequence, they kind of twist to the other side. When they're not, they twist to the B-DNA side. That's how that goes. Okay? Minor difference, super important to a molecular level of size, important to understand how things work, and so that goes. The RNA, as I said, has double OH, but their backbone is very similar to the DNA. Even the connection of phosphorus is very similar as well. As you can see, instead of a thymine, has a uracil in their position. Okay. And while DNA, they do form double-stranded helix, RNA, they can be by themselves, and they can twist and form double helix within themselves. They can get another RNA that's been produced that's similar. They can bond together to stabilize their molecule. RNAs are inherently stable, and this is important to make it easier to go to ribosomes and produce proteins, or to be folded into other components that have functions, like RNA that can transport substance, or RNA that can regulate parts of the DNA microcosm. Okay, the process of formation of RNA from a DNA strand is called transcription, and next class we're going to be talking a lot about transcription next Tuesday. Okay. And as I said, because this structure of RNA is very unstable, it can form hairpins, can form all sorts of different formats, and this way it has different functions as well. So while DNA is just a blueprint for a lot of different stuff, RNA can be a blueprint and as an intermediate between a DNA and a protein, but can also form structures that are going to have importance in transporting molecules or mostly transporting molecules. That's the second most important part of the RNA. So one that's important is the transporter RNA or tRNA. We have ones that are very specific. So we have a tRNA that is important to carry some amino acids, in this case phenylalanine. And there are some people that they don't produce this specific tRNA and they cannot taste phenylalanine, can even get intoxicated by it. And lastly, let's summarize the difference between DNA and RNA. So, while DNA, there's a deoxy, it's missing on oxygen in the ribose, RNA is not. DNA has the bases ACTG, while RNA, instead of a T, has a U, a uracil. In general, DNA has two strands when they're forming. It's very important to stabilize the molecule. RNA is formed as just one strand, and this strand can become a double strand later or coil by its, or bond by itself in the hydrogen bond. Okay. The base pairing is sequential, and a is going to connect with a T is going to connect with an a C with a G with a C. And that's the rule, and nothing changes that. Okay, that's how it goes, and every pair is going to be connected. There's no skipping, no nothing. Okay, while an RNA, adenine is going to connect with uracil, and that's a bigger difference. Okay, and they sometimes skip some pairs depending on what they need. The shape of DNA is a double helix, while RNA has a very variable shape and it's going to be according to its function. So there's a lot of different combinations. Okay, so that's that for today. I hope you guys enjoyed. Sorry again for not being able to be in class this Thursday, but I'm gonna see you guys on Tuesday. So I hope you guys have a wonderful first week of class, and see you guys next week. Okay, have a wonderful weekend. Bye bye.